In this video, we will look at the role of epigenetics in development. Epigenetics plays an essential role in our development as multicellular organisms. Development is the series of genetically programmed stages that take one individual from a fertilized egg to an adult, and through every stage in between. Many of the changes that we see in development are actually due to maintained by epigenetics. Here, we will explore three examples. The first example is genomic imprinting, which occurs during gamete formation and is the form of gene regulation in which the offspring expresses the copy of a gene from only one parent, because the copy from the other parent is epigenetically silenced. An example of this phenomenon is the LGF2 gene, in which only the paternal copy is expressed. This is due to methylation of the LGF2 gene in sperm, but not eggs. This methylation occurs at the imprinting control region and the differentially methylated region in this gene. But hang on, I thought methylation was only for silencing genes. Often this is the case, but not always. In this example, methylation actually prevents the binding of CTC binding factor, which itself inhibits transcription. Methylated copies of the LGF2 gene cannot bind CTC binding factor and can thus be stimulated by the nearby enhancer and be expressed. The second example is X chromosome inactivation. In female mammals, one of the two X chromosomes they have is inactivated in each cell. Sometime during embryogenesis, one X chromosome in each cell present at the time is selected for inactivation. X inactivation is controlled by two genes, T6 and XIST, found in the XIC region of the X chromosome. These two genes overlap, but are transcribed in opposite directions. The basic process of X inactivation is shown here, so let's walk through it. At some point during embryogenesis, the two X chromosomes in a cell pair up. These chromosomes have been expressing the T6 gene and have pluripotency factors, which facilitate that expression of the T6 bound to them. The pairing of these X chromosomes is facilitated by CTCF proteins. This pairing causes the CTCF and pluripotency factors to move to one of the X chromosomes, and this one will become the active X for that cell and all subsequent daughter cells. The chromosome without these factors now expresses the XIST RNA. Expression of XIST recruits proteins that spread along the length of the chromosome and begin to compact this chromosome. The end result is a highly compacted X chromosome called a bar body. All future cells that result from this cell division will have the same X chromosome active and the same one inactive. The last example is the formation of specific cell types in multicellular organisms. At some point during early embryonic development, a cell undergoes an epigenetic change, which marks it to become a specific cell type. In this cell, the tissue-specific genes are active for that cell type along with the general housekeeping genes. There are two types of competing protein complexes that regulate epigenetic changes during development and produce these specific cell types. They are the trithorax group proteins that are involved in gene activation and the polycomb group proteins involved in gene repression. Let's take a look at how the polycomb group complex proteins work to repress gene expression. The process is diagrammed in the figure here and involves two types of protein complexes, the PRC1 and the PRC2. Generally, what happens is the PRC2 binds to the polycomb response element in the DNA, and that leads to the trimethylation of lysine 27 on histone H3 in several subsequent histone octamers. This trimethylation prevents gene expression, but also likely recruits the PRC1 to bind to this region. PRC1 then acts to inhibit transcription in three different ways. The first is through chromosome compaction, where it causes nucleosomes to form a knot-like structure. The second is through modification of histone H2A by attaching ubiquitin molecules to it. And the third way is through direct interaction with transcription factors like TFIID, which are needed to initiate transcription.